One of the scientists to study DNA is Edwin Chargaff, and he figured out two important rules about DNA. Before we get into the rules, something that was already known about DNA is that it consisted of three parts. One part is a nitrogenous base, another part is a pentose 5-carbon sugar known as deoxyribose, another part of a nucleotide, which again, DNA is a polymer of nucleotides. The last part is a phosphate group. So those three parts make up DNA, repeating units to make up DNA. Now, something that he figured out in studying different types of organisms, different types of species, is that the DNA composition varied between different species. So a sea urchin had roughly 33% DNA composition, whereas a bacteria cell would have somewhere around 24%. So there was a difference in how much DNA different organisms had in their cells. So that was something that wasn't widely known. The other thing that he figured out was something known as Chargaff's rule. So he figured out that of the 4 nitrogenous bases, that certain ones occurred at certain percents. So for example, if a cell had roughly 32% A adenine, then there was roughly 32% thymine. If there was 17% guanine, then there was roughly the same percent of cytosine. And that again is known as Chargaff's rule. So what type of bond keeps the rungs together? It's a hydrogen bond. Again, the rungs being the nitrogenous bases. So a hydrogen bond is responsible for keeping the A's and the T's together and the C's and the G's. Now, it is hydrogen bonds, but you should know how many hydrogen bonds keep those molecules together. So adenine and thymine are held together by a double hydrogen bond. So there's 2 hydrogen bonds keeping those molecules together. Guanine and cytosine are held together by 3 hydrogen bonds within their molecules. Something you will hear a lot in studying DNA is the word antiparallel. DNA is an antiparallel molecule, meaning that they don't run exactly with the same orientation, with the molecules facing up or facing down. They're opposites of one another. So one end piece of DNA on one side, you will have a 5' end. So try to remember that the prime numbers have to do with the numbering on the sugar molecule. On the same side, like the left side, if you look at this picture, you see there's a 3' end. Again, having to do with how those carbons are numbered in that sugar molecule. So on the left, 5' at the top down to 3'. So if you look at the opposite side, you have now the 5' end is at the bottom and the 3' end is at the top. So they are opposites of one another, or they are anti-parallel. Now, how exactly can you remember this? Whenever I look at a picture of DNA, I look at the sugar molecule, again, a deoxyribose, a pentose 5-carbon sugar. So if you look at that blue molecule on the left, you see the oxygen in that carbon or in that sugar molecule. It's facing the top. The top oxygen is like an arrow. It looks like it's pointing upwards, at least to me, and that's the 5' end. The oxygen of the sugar always points to the 5' end. So if you look now at the bottom right, you can see that oxygen in that pentose sugar is now facing down. Well, it's also facing down towards the 5' end. I don't know if that helps, but that's a little bit of how I remember. which end is 5', and then you just automatically know the other side has to be 3'.